Well, hello, my fellow pre-meds. Today we have a very dense slide deck for you, and that is the structure and function of lipids. A lot of concepts you have to know here, a lot of terminology. I'd highly recommend that you use our Anki cards that are associated with this slide deck to be able to memorize all of those terms for the long run, although I will try my best to help explain the underlying mechanisms in an easy to understand way. So again, they can stick for the long run. And I'm appropriately dressed as olive oil today. All right, let's get started. So brief introductions here. So lipids are a diverse group of hydrophobic and amphipathic molecules, primarily composed of hydrocarbons. So hydrophobic, we know what that means. Amphipathic is a new term, so that means that the same molecule is both hydrophilic at one end and hydrophobic at the other end at the same time. Because they're largely nonpolar, they're going to be soluble in nonpolar solvents and insoluble in aqueous environments. Lipids serve, their major roles are energy storage, membrane, structure, signaling, and insulation, as we'll see in subsequent slides. And these are the major categories which we will start discussing right now. So fatty acids are long chain carboxylic acids. So they are carboxylic acids, but they just have a really long alkyl chain. Now the major categories of fatty acids are saturated fatty acids and unsaturated fatty acids. So when it comes to saturated fatty acids, as you can see right here, they don't contain a double bond. And so every single carbon over here, with the exception of the first and the last one, are bound to two hydrogen atoms. So that means they're saturated with the maximum number of hydrogen atoms that they can have. Whereas when it comes to the unsaturated fatty acids, as you can see, they have a double bond, and therefore the carbons that are participating in that double bond will be missing one of those hydrogen atoms. And by the way, when you have a saturated fatty acid, it's easier for the molecules to stack up on one another, and so therefore they'll be more stable. So they will have higher melting temperatures. So essentially, at room temperature, you'll see that saturated fatty acids will be in the solid form, such as butters, whereas when it comes to unsaturated fatty acids, because they'll have lower melting points, so they'll melt at lower temperatures, like at room temperature. So you'll see that oils are liquids at room temperature, and so oils will have unsaturated fatty acids. Now, when it comes to unsaturated fatty acids, we have two types. We have the cis unsaturated fatty acids and then the trans unsaturated fatty acids. So notice that the trans unsaturated fatty acid is more linear. So therefore it's more stable. So again, it'll contribute to a higher melting temperature. It'll make them more stable. And when it's present in a plasma membrane, as we'll see in subsequent slides, it's gonna reduce the permeability or the fluidity of that membrane and increase rigidity, whereas the cis bond will increase fluidity and decrease rigidity. So the reason why is because if you take a look at the cis double bond, you have the bulky groups on the same side of that double bond. If you remember, that's what cis means from our organic chemistry. And these will have a more difficult time stacking up on top of one another compared to the trans fatty acids. All right, and then free fatty acids, again, because fatty acids are hydrophobic, they can't really circulate on their own in the blood, so they circulate bound to albumin non-covalently. All right. Oh, and just one note here on terminology, on naming of fatty acids. This sometimes shows up on the MCAT. So you name it based on the number of carbons and double bonds. So the first number will be the number of carbons. Second number will be the number of double bonds. So if you have 18 carbons and 2 double bonds, of course, other than the carboxylic acid double bond, then you would say 18-2. And then one thing I didn't write down here because it's really not high yield, but it's worth knowing. So when we talk about, for example, omega-3 fatty acids, if you're familiar with that, from like they're healthy for your brain, the omega is referring to this part right here, and that 3 number is referring to how far the first double bond is from that omega part. Okay, so now let's talk about triacylglycerols, sometimes called triglycerides, and they're named based on their structure. So if you take a look at their structure right here, you have 3 fatty acids and 1 glycerol. A glycerol, notice, is an alcohol or a molecule that contains 3 alcohols And then once these combine with one another via an esterification reaction, because recall when you have a carboxylic acid, which is what fatty acids are, interact with an alcohol, that is an esterification, and you're going to form an ester where you have a carbonyl bound to an oxygen on one end and then an alkyl chain on the other end. So essentially what you have is 3 fatty acids. When you form that ester, we call them acyl. So instead of fatty acids, we call them fatty acyl. So you have triacyl, 1, 2, 3, and then glycerol, which is this part over here. All right, so they serve as a major form of storage, like energy storage in the body. And because they're highly nonpolar, they are typically insoluble in water. And now you can hydrolyze them, also like in a process called saponification. So this is base catalyzed hydrolysis, and it'll form soap, and we'll see that right here. So this is the ester hydrolysis of triglycerides using NaOH, which is a strong base. Essentially what you have is each one of these NaOHs is going to hydrolyze one of these esters to revert them back to their carboxylic acid form, or in this case carboxylate, the ion, and the alcohol. And then notice how now these free fatty acids have long hydrophobic chains and then a hydrophilic carboxylate group over here. And this is essentially what soap is. So this is how they make soap. Essentially, like let's say you're trying to wash a greasy pan or an oily pan. When you're adding water to that greasy pan, water and oil will not mix. But if you add a soap, the reason why they then mix and you can clean that greasy pan is because the hydrophobic tail over there will interact with the oil, and the hydrophilic carboxylate ion will interact with the water, and then it'll cause them to mix. All right, now let's talk about phospholipids. So phospholipids, again, don't be confused by this. It's very simple once you break down the structure of it, which we can see right here. So you can see that phospholipids, and we'll go through these points in just a bit. So phospholipids are very similar to triacylglycerols. So we said earlier that triacylglycerols are formed by a glycerol and 3 fatty acids, right? If you look at phospholipids, well, it's essentially the same thing for the first part, which is the glycerol. You can see the glycerol right here. And then the 2 fatty acids, you can see 1 fatty acid and another one over here. But then instead of the third one, we've just replaced that third one. So I've color-coded those carbons. So this yellow carbon is essentially the same as this one. Instead of it having a fatty acid chain over here, it has a phosphate-containing head group. So now that is the structure of the phospholipid. Now, phospholipids, it's important to note that they are amphipathic. So the phosphate-containing head group is going to be hydrophilic, so it's going to interact with aqueous environments, whereas the fatty acid chains are going to be hydrophobic. Phospholipids are the primary structural components of the cell membrane. Again, we'll revisit this in the next slide again. So they form the plasma membrane. In aqueous environments, phospholipids spontaneously form a bilayer, and we'll see how this works in a bit. Finally, if you have unsaturated fatty acids in a bilayer, as we mentioned earlier, they're going to increase fluidity, and that's because unsaturated fatty acids means that they have a double bond, which means that they're not going to stack up as neatly on top of one another, and therefore they're going to increase the fluidity of the membrane. So you can see this is the formation of the bilayer. Essentially, if you throw in a bunch of phospholipids in an aqueous environment, such as in the body or in a test tube, they're going to arrange in this fashion. So they're going to arrange so that the hydrophobic tails are going to want to escape the aqueous environment, and they're going to clump up with one another, and then these phospholipid heads are going to be able to interact with the aqueous environment, so they're going to go on the outside. Now notice over here how this is an unsaturated fatty acid, whereas this one's saturated. So this one's going to increase membrane fluidity. In other words, it's going to increase membrane permeability. And then this is just a more complex structure of the plasma membrane. We're going to talk a lot about the different proteins, glycoproteins that can exist within the plasma membrane in a subsequent slide deck in the future about the plasma membrane, because there's a lot to talk about over there. All right, now let's briefly talk about micelles and liposomes. So again, these are very similar to how the bilayer forms. Essentially, they are phospholipids. So you can see that over here, this is what we saw in the previous slide with the formation of the bilayer. Well, a liposome turns out to be just the spherical form of that. So this is a bilayer sheet. Liposomes are a spherical bilayer. And then micelles are a bit different. So they don't have a bilayer, they just have the surface is hydrophilic, the hydrophilic heads, and then the core is purely hydrophobic. Now, these are amphipathic because they contain the hydrophilic heads and the hydrophobic tails. When it comes to micelles, they're primarily involved in the digestive tract. So in the intestinal lumen, the environment is quite aqueous. So for those fat-soluble vitamins, as we'll see in a bit, and for fatty foods, you need something to be able to interact with them, and that's going to be the core of these micelles. So you'll find the fat-soluble vitamins in the core over here, and then the hydrophilic heads will interact with the aqueous environment. Liposomes are sometimes involved in membrane transport or drug delivery. So again, the difference between micelles and liposomes is that the core in the liposomes is actually hydrophilic because you have a bilayer. So hydrophilic drugs will be in the core over there. Hydrophobic drugs will be interacting with these fatty acid groups over here. Okay, now waxes, again, this doesn't show up a ton on the MCAT, but it's worth knowing because it's pretty straightforward. So waxes are essentially an interaction between a long chain fatty acid and a long chain alcohol. And guys, I can't emphasize it enough how important it is to understand what an esterification reaction is in the context of fatty acids and just lipid discussion in general, because it shows up a ton. So essentially in organic chemistry, when you react an alcohol with a carboxylic acid, you get an ester. And an ester is this structure right here where you have a carbonyl bound to an oxygen and then followed by like an alkyl group, okay? So what we're looking at over here is our long chain fatty acid. This part is our long chain fatty acid. This part is our long chain alcohol, okay? Now because they've already interacted, you can't see the OH part of the alcohol. Now, these are extremely hydrophobic, so they assist in like waterproofing plants or animals, so that plants, for example, don't evaporate their water, and then animals, sometimes, you know, they have hydrophobic coating. So, for example, ducks, when they're on the water, they won't get wet. So that's where waxes come into play because they're extremely hydrophobic and water resistant. Now, terpenes are important insofar as they're involved in the synthesis of these more important biological molecules over here. So this is the structure of an isoprene, which is the functional or the most basic unit within terpenes. Essentially, the way you want to name terpenes is that if you have two isoprenes, you have the most basic form, which is a monoterpene. If you have 4, then you have a diterpene, and then if you have 6, so you're increasing by 2 every time, you will have a triterpene, and so on and so forth. So a tetraterpene would be 8 isoprenes. Now again, isoprenes are involved in the synthesis of steroid hormones, cholesterol, and vitamin A, which we're going to see all of these in subsequent slides. So steroid and cholesterol. Steroids and cholesterol are extremely high yield on the MCAT, and you know, steroids are not just like anabolic steroids, you know, the ones that bodybuilders will sometimes abuse. Steroid hormones are involved in, you know, your sex hormones, in the regulation of the salt and water, you know, your blood pressure in the case of aldosterone, for example. But more on that when we get to our biology playlist, you know, the endocrine system. So steroids are lipids characterized as 4 fused hydrocarbons. So you can see that structure right here. This is like the most basic fundamental structure. Typically, there will be 3 cyclohexanes and 1 cyclopentane. And then steroids typically act as signaling molecules that regulate gene expression and metabolism. And again, more on this when we talk about in our biology playlist. But for here, I will just mention that like a significant difference between steroid hormones and like peptide hormones, that is hormones that involve either proteins or amino acids, is that the ones that contain proteins or amino acids are typically involved in short-term effects, such as opening an ion channel, whereas steroid hormones are more involved in long-term effects, such as regulating transcription and expression of genes. Okay, so now let's move on to cholesterol. So cholesterols are also amphipathic, like we saw with phospholipids, and that's because not only do they have this hydrophobic side over here, but they also have the hydrophilic OH. So they are amphipathic. They can interact with both aqueous and hydrophobic environments. And then one important point that does show up quite frequently on practice exams is that cholesterol will maintain membrane fluidity. But unlike when we talked about you know, fatty acids, we talked about how if you have a saturated versus unsaturated fatty acid, the saturated, which doesn't have double bonds, will increase rigidity, whereas the unsaturated will increase fluidity, right? Whereas cholesterol sort of plays both roles. So it can increase fluidity when appropriate. So like, let's say in cold environments, that cell membrane is going to want to crystallize and become very rigid. Cholesterol can help increase fluidity, whereas in really warm temperatures and hot temperatures, it will want to decrease fluidity so as not to, make it too permeable. So it plays both roles, and that's important on the MCAT. And then cholesterol also serves as a precursor for some steroid hormones, bile acids, and vitamin D. And then that's just the cholesterol molecules embedded there within the plasma membrane. So you can see how the OH is interacting with the aqueous environment, whereas the other part of it, which is the hydrophobic part of it, is interacting with the fatty acid tails. Okay, so these are some examples of steroid hormones. Again, a lot more on these when we talk about our biology playlist, or in our biology playlist. Progesterone, typically associated with the female sex. Testosterone, typically associated with the male sex. And then aldosterone, involved in regulation of salt reabsorption and water for blood pressure regulation. Now, when it comes to fat-soluble vitamins, so recall the water-soluble vitamins are vitamins B and C. We're not going to discuss them over here. For the fat-soluble vitamins, you don't have to know a ton about the functions of these. A lot more on that when you're in med school. But for our purposes, for the purposes of the MCAT, you should know like the very broad function of each of them. So vitamin A is involved in vision and cellular differentiation. Vitamin D regulates calcium and phosphate homeostasis. And again, more on that when in our biology playlist. Vitamin E functions as an antioxidant that protects cell membranes, and then vitamin K is involved in coagulation. And the way a lot of students remember vitamin K is like vitamin K coagulation or clotting factors, even though those start with C's, but it sounds like it's a K, so you can memorize it like that. And then vitamin A over here is, so recall how we mentioned that isoprenes are the precursors for this. So notice our isoprene structure over here is very similar to the structure that we saw right there. And then we also have vitamin D over here, which was the synthesized from the precursor cholesterol. So there is some similarity over there in terms of, you got a cyclohexane, and cyclopentane. Okay, let's move on to prostaglandins. I believe this is the last slide. Yes, last slide. Okay, so prostaglandins, again, not very high yield on the MCAT, but I thought it was worth knowing just so, you don't want, the reason why even the low yield topics I would sometimes bring them up, because I don't want you to be surprised with something on the MCAT, like the first time you see something is on test day. I think it's worth just briefly mentioning them over here. So prostaglandins, what you want to know is that they are lipids. They're involved in signaling for like inflammation, pain, fever, and smooth muscle activity. And then this, while you don't necessarily need to know that aspirin is inhibiting the cyclooxygenase enzyme, I think it's worth, just knowing that fact. It's an easy fact that might show up in passages. And then they'll ask you questions like related to it that are fair game on the MCAT. So, for example, the fact that they are lipids. And then the inhibition of COX is going to reduce prostaglandins. And just to tie into enzymes, this is an irreversible inhibitor, aspirin, because it will irreversibly inhibit cyclooxygenases. So the inhibitor is not going to wear off. You need to, essentially, the body needs to express new cyclooxygenase enzymes. All right, this was a dense slide deck, a lot of terminology, a lot of memorization. Be sure to use our Anki cards associated with the slide deck, or even create your own by using the slide deck. So like download the PowerPoint and then create your own if you feel like that's more helpful for you. And yeah, if you have any questions, you know where to reach me, and go ahead and crush that MCAT. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it.